Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 161 for the podcast. Alnylam was the very first company to translate RNAi from Nobel Prize winning science into a commercially scalable platform. Founded in 2002, the company today has 5 different RNAi therapies on the market and a pipeline that continues to expand. While their early focus was on rare disease, today they are expanding into more common conditions, partnering with top pharma companies to bring next-generation therapies to market. This week, I sat down with Paul Nioi, Senior Vice President of Research at Alnylam. Paul has more than 20 years' experience in biotech and biopharma, and he walked me through Alnylam's mission and place in the biotech ecosystem, the science of RNAi, the early success treating metabolic disease, and exactly where their platform is expanding into now and in the near future. Paul explains the capacity of RNAi to address neurological and infectious disease, the value of large-scale genomic initiatives, and how he sees drug discovery evolving in the years ahead. I hope you enjoy my talk with Paul Nioi of Alnylam. Paul Nioi, welcome to Beyond Biotech.
Hi, Dylan. Great to be here.
Paul, let's get started by just learning a little bit about you. How did you find yourself here at Alnylam? Tell me a little bit about your journey in the biotech industry.
So I've been in the industry for over 20 years now, and I was always from the very beginning when I started my academic career, really determined at some point to make the transition into industry. In fact, I did that as a postdoc. So I worked for a company called Shearing Plough for a number of years, first as a postdoc, then as a scientist, really focused on small molecule drug discovery. And I became fascinated with the challenges that existed. When I joined Shearing, I was so naive and thought that it's straightforward. You understand a bit of molecular biology, and quickly realized how complicated the business is. And what really struck me was like how many drugs fail in the clinic and the rate of failure. It was just incredible. So that kind of became a focus for me. How do we do a better job of finding good drug targets that can actually work when you advance those molecules into the clinic? And really became focused on new technologies that we can use to solve that problem. So I was at first focused on using transcriptomics and microarrays and different ways of profiling gene expression in a very broad way, which it was trendy at the time, and it was kind of thought, well, maybe if you take this unbiased view of disease, you'll come up with better drug targets. And I joined Amgen in 2010, who were very invested in this approach, and continued on that journey, using these technologies to help find better targets. And somewhere along the way, I became less enamored with transcriptomics and more fascinated by human genetics, which to me was just an easier way to look at the problem. If you find humans that lack a gene and they have a certain phenotype, it's so clear that that gene is involved in that disease, and the target could be a really powerful, meaningful target, because that protein is doing something in the disease. And so that really became the focal point of my career. Amgen acquired Decode Genetics in Iceland. I was lucky enough to spend some time in Iceland with Kari Stephenson and the team there, who really taught me the nuts and bolts of how you study population genetics to find drug targets. And it was a great experience. I came back to Amgen at the Boston site and kind of was bridging what they were doing at Decode with the mothership at Amgen, trying to come up with new targets for different disease areas. We had a lot of success in finding some exciting new things that made it into the pipeline. But there were many things that we found that weren't druggable. And that's when I really became fascinated by what Alnylam was doing, because they had this amazing technology where any gene in the genome was druggable with RNAi. You could knock down the expression of any gene. So this question of can we drug it with a small molecule? Is it soluble? Can we go after it with an antibody? Didn't matter anymore. And so I ended up joining Alnylam mid or early 2018. Um, and I have to say, all of my kind of the hype I had in my head about it was not even close to how amazing it really was. When we get it, really is true. You can knock down the expression of any gene in the genome. And the amazing thing about the platform is being able to go from an idea to filing a CTA in as little as 18 months. It's an amazing, amazing platform. And so since then, I've as been in a couple of different roles since I've been here, but now I lead essentially the drug discovery research organization. So everything from figuring out new targets using genetics to the early target validation to leading the teams that ultimately find the drug that we're going to then advance all the way into CTAs and into the clinic. And it's been an amazing journey.
You mentioned a couple of different companies there, from Sharing through Decode, Amgen as well now,
Yeah.
to Alnylam. But you also work with the Rett Syndrome Research Trust. Does that complement what you're doing at Alnylam?
It does, yeah. So I got involved maybe 5 years ago now. And this is, as you may know, is a devastating disease caused by loss of function in a gene called MECP2. And we at Alnylam had recently established a way of delivering our therapies to the CNS. And so we'd been really kind of investing in thinking about neurological diseases. Rett syndrome, unfortunately, is not a disease that's amenable to RNAi. It's caused by a loss of function in a particular gene. But there's two things there that really I think have been very valuable for me. One is um, you're so close to the patient journey when you're involved in a research trust like that, that it really brings it home to you, not just ideas for being able to tackle the disease, but the impact that it has on the patient and on the family and on the caregivers. It's really, really eye-opening, and it gives you a healthy appreciation for, this is why we're doing what we're doing. The other side of that is that there are many other technologies and approaches that we may use to try and intervene in Rett syndrome, everything from gene editing to gene therapy. There's a whole host of ideas, and so it provides a lot of exposure to what others are doing in the field and complementary technologies to RNAi that may be used for other things, which again is very helpful.
Tell me a little bit about Alnylam. What's the mission of the company? What are they setting out to do?
So we were founded in the early 2000s with the sole purpose of turning RNA interference into a medicine. So this was a discovery that was first observed, believe it or not, in plants and petunias, where researchers introduced double-stranded RNA that they thought were going to make the flowers more purple in color, and they ended up making them white. And there was this mystery about what was going on, and it took several years and more experiments in C. elegans to figure out that actually there's this natural process that exists in these organisms where if you introduce double-stranded RNA, there's a particular protein complex called RISC, or RNA-induced silencing complex, that unwinds the two strands. It takes one of the strands, and then it floats around the cell and it finds sequences that are complementary to that little piece of RNA that it's found and chops it up. It stops it from being expressed. So it's a way that the cell can protect itself from foreign invaders, from viruses and so forth. And it turns out that mechanism exists in mammalian cells and in human cells as well. So there's a famous paper from the lab of Tom Tuchel where he showed for the first time that this works in human cells and that this complex exists in human cells. And that was the piece of data that really was the foundation of Alnylam. It led to the founding of Alnylam because we knew this is a mechanism that exists in human cells. And wow, what if you could introduce these RNAs into cells? You could drug anything. You could knock down any gene in the genome. So our mission has been, how do you go from that amazing academic work that actually led to the award of the Nobel Prize to Fire and Mello? How do you turn that into medicine? How would you use it as a medicine? So RNA, double-strand RNA is extremely unstable. If you introduce it into the bloodstream, it gets chewed up. There's so many enzymes that exist that will just degrade it immediately. Not only that, it's very charged and it doesn't get across the membrane of the cell. So you can't actually, it's unstable and you can't get it to where it needs to be, which is inside the cell. So for really the first decade of Alnylam's existence, it was really a mission in how on earth do we tackle those two fundamental problems. We came up with a way of stabilizing the RNA by introducing modifications on the backbone of the sugars that make up the RNA molecule that stop it being recognized by endo and exonucleases, so it's stable. And then we came up with two different ways of actually getting it across the membrane of the cell. One is to use lipid nanoparticles, which you're probably familiar with from the COVID vaccines and so on, where you package the RNA inside these lipids, and those lipids bind to proteins in the blood that are taken up by the liver, and the payload is delivered into the liver. So that's one approach. But we found a more elegant approach, which is to add what we call conjugates, or a sugar molecule for targeting the liver called GalNAc that we put on one end of the RNA, and it's recognized by a receptor on the surface of the liver that then endocytoses the drug, and it's actually stored inside the cell and gradually leaks out into the cytoplasm and gives you knockdown over a prolonged period of time. So now we've gone from these very early challenges to drugs where we've got this profile where they're very stable. You can dose subcutaneously once every 3 to 6 months, and you can get a pharmacodynamic effect that lasts literally months. It's incredible. It's like a really amazing profile because it can mean that the patient doesn't have to remember to take their pill every day. They just receive an injection infrequently. And our platform initially focused on targets that are in the liver. We've had several drugs now approved that use that mechanism, and we've since expanded to deliver our medicines to the central nervous system. So we have several programs in the clinic now in the CNS. And our goal is by 2030, we're going to be able to deliver siRNA to all major tissues in the body.
I want to ask you a little bit about the partnerships that you've got, because as I can see, you've got a couple of different sorts of partnerships. The first are with these sort of genetic validation, these human genetic banks, UK Biobank, Our Future Health, Discover Me South Africa. And the second sorts of partnerships are with big industry players, the Roche, the Regeneron. How do these different partnerships position Alnylam in the market. How does it position you within that broader biotech ecosystem and reinforce what you're doing?
Yeah, I mean the two different types that you mentioned are they really serve different purposes. So we can maybe start with the genetics partnerships that you brought up, where I mentioned to you earlier about kind of the challenges in drug discovery, where you're really trying to come up with what's the best target and how do we understand targets? How do we understand human disease? And using human genetics is a very powerful way of doing that, because excuse me, someone has a mutation in a certain gene, and they have a certain disease, or they're protected from a disease. That gene's involved in that disease, for a fact, in humans. So that really helps us to understand where to point our technology. What are the opportunities for RNAi where we find that individuals that lack a given gene are protected from a disease? Those are perfect targets for RNAi. And can we mimic that phenotype in the whole population with our medicines? So we've got some really powerful examples of things that have come out of UK Biobank, for example, where we found this target called INHBE, where individuals that lack one copy of that gene are protected from metabolic disease. On the topic of the genetic partnerships, as I said, you have a very powerful lens that you can find targets through using genetics. It's really a case of being able to find individuals with interesting phenotypes in the data that tell you this gene is really important in this disease. And it might be, something where we say, people that lack this gene are protected from a disease, and we can mimic that phenotype by knocking down the gene with siRNA. That's the perfect scenario. In other cases, you're really learning about the fundamental causes of a disease, which is often mysterious in the literature, and it kind of gives you the information to help you think about therapeutic approaches that may be able to help. That data is something that we've been able to access through collaboration. So, for example, with UK Biobank, we partnered with them and a number of other pharma companies to sequence the exomes of that whole cohort. So there's 500,000 people in the UK. We generated the sequence level information, and we had access to all of the medical records going back decades for those individuals. So it's an amazing resource to be able to ask the types of questions that I mentioned. Same with Our Future Health, which is an even larger version of UK Biobank, where we aim to recruit 5 million people in the UK. And then adding data from more diverse populations. So Discover Me South Africa, we're trying to recruit 100,000 people there. And of course, add a whole different lens of diversity that we don't have in the data from UK Biobank or Our Future Health, which is predominantly European. So those are really partnerships that help us make discoveries, help us figure out what's the right target for our technology. The other types of deals that we've done are later stage. So Regeneron is a company that we partnered with in 2019, I think, where we had just figured out how to deliver our medicines to the brain with a novel conjugate and through intrathecal injection. And at that point in Alnylam's kind of existence, we were, of course, excited about the implications of that and the ability for us to be able to really make an impact, hopefully on severe neurological diseases where there's huge unmet need. But you need to have a good partner when you're at that kind of stage as a company where we didn't have any meaningful kind of money coming in the door, so we needed a partner to really help us be able to take these things into the clinic and see what the promise was, and can we really help patients here. And so we've had a great partnership with Regeneron, and that's really enabled us to work with them together to advance a number of different programs into the clinic. And then even later stage than that, we have a relationship with Roche for our hypertension or potential hypertension medicines, albiceran, where we're co-developing that drug, and that's moving into phase 2. Well, it's in phase 2 trials. We're doing an outcomes trial. These are very big cardiovascular trials, and those are again areas that were new for Alnylam, but huge potential for that as a medicine, and we needed a partner to really help us bring that forward. So those are some examples of where those things have really aided what we do.
Let me ask a little bit about a medicine that's already out there, that's already approved on Patro. This was your first approved RNAi drug, and it targeted, and I hope I get this right, hereditary transthyretin-mediated amyloidosis, which is a rare genetic disease, or would be if I can pronounce it correctly. What were the challenges of developing that drug, particularly around really getting the RNA stable and getting it delivered?
Yeah, so you're right. Patisiran was our first drug that was approved, and it's an siRNA that knocks down the expression of transthyretin, or TTR. That's a protein that's produced by the liver. I'm always amazed by the liver. The biology it's all over the place. It makes proteins that involved in blood clotting, involved in the immune system, and then TTR, which is something that's secreted, but actually in people that have certain mutations in that gene, the secreted protein unfolds and it aggregates and forms this amyloid-like structure that gets stuck in the extracellular matrix of certain tissues, predominantly peripheral nerves and also the heart. And so patisiran was designed to knock down, silence the expression of TTR in the liver and stop it from being made, and then hopefully help with the manifestations of the disease that I mentioned. And it's very rapidly progressing. It's something that at that point there really weren't any treatments out there for these patients. So our approach was really, okay, well, we know this protein is involved in the disease. If we can silence it, will that work? Will that have an impact on the disease? We really had to tackle the fundamental challenges that I mentioned earlier in the interview, which were, how do we stop the siRNA from being chewed up by nucleases straight away? And how do we get this thing across the membrane of the hepatocyte and into the cytoplasm so it can actually affect gene silencing? We first took a chemistry approach to understand, I suppose, mechanistically, how are nucleases actually recognizing the siRNA, and which particular sites on the siRNA are susceptible to being degraded? What are these enzymes recognizing? And we discovered that if we introduce specific modifications on certain positions on the backbone of the siRNA, we could actually prevent that degradation. We could prevent the siRNA from being degraded, or at least greatly reduce the degradation. So that was one fundamental discovery. The other was that if you package the sRNA into these lipid bubbles, you can protect them even more from degradation, and those allow delivery to the liver. But it took a huge amount of iteration to actually get to what I'm describing. What I just told you in 2 or 3 sentences was literally 10, 15 years worth of work to really understand fundamentally what was going on and be able to engineer out, these problems that existed. And so that was really the journey that led to patisiran. At the same time, no one had ever made sRNA as a drug. No one had made lipid nanoparticles on that scale to be used in humans. And so we had to figure out how to make these things and how to actually do it in such a way that you could use them in humans. And how do you administer them? How do you make sure that patients are not having untoward reactions to these LNPs. So there was a huge amount of work that really went into the clinical development and the manufacturing to really bring it all the way through. But it was really a very powerful program, because in the context of that program, we solved so many problems that then led to all of the other medicines and potential medicines that are in our pipeline. And so we're now at this sort of inflection point where we've solved a lot of these platform issues, and the technology is really poised to take off. I think in the same way that we've seen with monoclonal antibodies, where there's a few examples right at the beginning where people managed to engineer these things and they ultimately got to the point of being a drug. And now we've seen this absolutely massive explosion in the number of monoclonals that are out there and that are in development. I think we'll see the same thing with siRNA.
Let's talk a little bit about that pipeline, because initially Alnylam was really focused on rare disease. Now it's starting to expand into more common conditions. You mentioned hypertension before, maybe even into Alzheimer's as well. What's enabling this shift? Is it just getting the platform right, and now you can apply that to different targets?
I think there's a few components to this. There's always risk when you're developing a drug, as you well know, a very high rate of failure in the industry. So for us, when we were first thinking about translating this technology into the clinic, we really had carried 2 risks. One risk is the platform itself. Is it going to work? Are you going to be able to protect the sRNA from degradation? Is it going to get into the hepatocyte? Is it going to silence the gene in question? And the other risk is the target, right? Even if you knock the target down, is it actually involved in the disease? Is it going to have an effect if I knock this thing down? So we really gravitated towards monogenic diseases where there was a lot of unmet need, but the target was crystal clear. We knew that gene, so we've talked about TTR. We knew TTR was involved in TTR amyloidosis. It's the cause of TTR amyloidosis. So it's a really, I think, powerful way of not only focusing what you're doing on an area of unmet need, but balancing the risks that you're taking from platform with and target, and saying, well, I'm really going to focus on these targets because I know that fundamentally this just makes the good biological sense. Plus, these are clinical trials that we can wrap our head around. It's not a gigantic indication where we're going to have to do a cardiovascular outcomes trial with 20,000 people or something like that. Something that's manageable for a company of Alnylam's size at that time. And that really allowed us to establish that this is a new class of medicines. This is something that actually works, and we've seen very positive data for in the clinic. And now we're thinking about, well, if it works here, where else could it work? And what's the world of targets that we could pursue? What are all of the possibilities that we could go after? And it turns out, even if you just think about what we can do in the liver, again, the liver is involved in so much of the physiology of the body. We're not just talking about treating liver diseases. There are so many opportunities, from lipids to blood pressure to immune system to rare TTR polyneuropathies to uh, porphyria, primary hyperoxaluria. These are areas that we focused in. So it's very broad, and we're really now looking at the opportunity of this. This is a whole new class of medicines, and it's something that could be used to help millions, potentially millions of patients around the world. And we really want to be able to explore the full potential of the technology in terms of tackling some of these areas of huge unmet need in society. Hypertension is a great example there. It's one of the biggest challenges that we have in managing patients that have hypertension. It's a major contributor to cardiovascular mortality. Alzheimer's disease, as you know, is an area where there's just massive unmet need, and there's so much that we hope we're able to do there. Huntington's disease is another example where we'd love to be able to really make an impact, and we think we have a technology that can. So I think it's really about, we've proven the platform, and now we have a very, very much a patient-focused approach, and we want to make sure that while we're at this inflection point from the technology, we're pointing it at diseases where we think we can really make an impact, and there's a huge amount of unmet need.
Let me ask about one of those areas. You talked about the importance of TTR, and once you understand the role that plays in the disease and you can target it, what about the discovery of INHBE mutations using that biobank data that you talked about before, the partnership there? How is discovering that sort of mutation affecting the way that you're working on cardiometabolic disease?
So this was a discovery that we made, as you mentioned, through UK Biobank. We found that individuals that lack one copy of this gene INHBE are metabolically very healthy. They actually have a smaller waist circumference, so they have less fat around the middle of their body. And that fat that's kind of stored in the center of the body is actually very harmful. It's called visceral fat. Secretes a lot of pro-inflammatory factors. It's just something that you really don't want to have. And so INHBE, people that lack INHBE seem to have less of that fat. And the knock-on consequence of that is they have lower LDL, they have lower blood pressure, they have lower glucose in their blood, they have lower risk of type 2 diabetes, they have lower risk of cardiovascular disease. So there's this whole panoply of cardiometabolic benefits in these people. We also found, by the way, that people that lack the receptor for INHBE, called ACVR1C, are similarly protected. So we had these two pieces of genetics that said if you lack the ligand, you're protected. If you lack the receptor, you're protected. So it's a very compelling story. It was completely new biology. This was not something that was out there in the literature that you could say, oh, okay, INHBE does X, Y. It wasn't really characterized. And what the biobank gave us was, we were the first to ever publish that. We got the first insight through that data to say this is an important gene in this disease. And it led to a program at Alnylam that we're now advancing towards the clinic with the hope of being able to mimic what I just described to you, which is this sort of metabolic benefit that these people have. Imagine if we could do that with an siRNA and give that benefit to a broad population. So we're really excited about moving that program forward.
I guess the other thing you're working with is with Roche. You mentioned that you're targeting hypertension with them. How does that actually differ from conventional antihypertensive drugs? That in terms of their mechanism, in terms of the way they're administered, what makes them different?
So this is a medicine called zilebesiran, which is in development for the treatment of hypertension with, as you mentioned, with our partner Roche. The target of zilebesiran is a protein called angiotensinogen, which is made in the liver. It's kind of a protein that's central to control of your blood pressure. It's secreted by the liver, and it acts on the walls of blood vessels to cause them to constrict and tighten. There are other ways of drugging this pathway that people have gone after the receptor or enzymes that process AGT as ways of being able to treat blood pressure, but we act upstream of all of those medicines in a way that you could think about it like turning off the tap. So if you have a tap that's in this gradual sort of drip, drip, drip of angiotensinogen being produced, you can either try and mop up the water mop up the floor as it's being produced, which is essentially what all these other small molecules are doing, or you can turn the tap off, and we aim to turn off the tap. That's our focus. So we silence AGT, and we have a really, really amazing profile with this drug, where it's given infrequently, once every 6 months, and you get this clamped pharmacological effect. Whereas, if you look at people's blood pressures when they're taking small molecule inhibitors of various enzymes, they have an effect for a period of time, and then as the day goes on, that effect wanes, and so the blood pressure will at some point be lower, at some point it will return to baseline, and you get this variable response. When we've looked in our clinical studies at zilebesiran, we have a clamped effect, so the blood pressure stays low over the whole day because we have this months long duration of action. So it's a really, really interesting profile, and a little bit like LDL. I think that the determinant of whether ultimately you're going to have cardiovascular events because of your high blood pressure or high LDL is a result of the area under the curve. How long were you exposed to a high level of LDL for? How long were you exposed to high blood pressure for? So we're really hopeful that clamped effect translates into something really meaningful in the clinic. And the one thing I'm not mentioning, which I think we all appreciate, is people forget to take their medicine every day. They're not compliant. So it takes that issue out of the equation because you don't have to remember to take it every day. You're having an injection infrequently that's going to give you that sustained effect. So this is all stuff we're really, we're in the clinic with this. We need to see what the profile is ultimately going to be of the drug, but we're really excited to be able to potentially bring something like that to millions of patients.
Let's take a look forward now. If you're thinking about Alnylam's RNAi platform evolving, and it continues to evolve, can it move into different therapeutic areas if we're looking 5-10 years ahead, neurological conditions, even infectious disease? Is there a chance?
There is indeed. I mean, we're already now in the clinic with a number of programs in the central nervous system. So we developed a new delivery approach that allows us to get our siRNAs into neurons, into cells of the brain. And one of our lead programs is called mivelsiran, which is targets the amyloid precursor protein, which is involved in Alzheimer's disease. So, I mean, that's one example. However, we are determined that by 2030, we're going to be able to deliver siRNA to every major tissue in the body. Not only that, but we have ways of delivering siRNA to multiple tissues at the same time. So you start to really open up the door to say, wow, if there's a certain gene in, I don't know, skeletal muscle or in adipose tissue that's a great target, we'll be able to do that. We'll be able to go after that. We'll be able to tackle that disease. Or maybe you need to be able to tackle, knock down, silence the gene in multiple tissues at the same time to have a therapeutic effect. And we have a technology that we believe is going to allow us to do that. So it really starts to open up all of these possibilities. And I mentioned, us being at this inflection point where we're able to really look forward and say, any disease where RNAi makes sense, we're going to be able to tackle, because we're going to have these delivery solutions to get the drug to where it needs to be, and be able to turn what was undruggable into something that's druggable. So that's really what we're excited about. And of course, infectious disease is one of those areas that we'd be able to explore. In fact, we've had a partnership with Vir in HBV as one example of where we've applied RNAi in infectious disease, and so these are the types of things we'll be able to do.
A couple of weeks ago on the show, we had Neil Ward of PacBio talking about the national newborn genome sequencing program that's going to be rolling out in the UK. How do you see the role of genetics and large-scale genomic initiatives like that, like Our Future Health? How are they going to shape the future of drug discovery for companies like yours and for others?
So I think there's several areas that they're going to impact. First and foremost, I've mentioned this several times, they are going to be extremely valuable in helping us understand the basis of human disease. For many, many years, we've used animal models to really try and understand human disease, mouse models of human disease. And we know from the failure rates of drugs in the clinic, those do not represent human disease well. They don't uncover the real core drivers of human disease. So genetics and population genetics in particular give you an insight that you can't gain from a mouse. They're really telling you about natural variation, nature's experiments, natural variation in a human that leads to a certain phenotype. And that opens our eyes to what's actually important and what the real targets are. So that part is going to be incredibly valuable. And the more that data grows, the more initiatives there are like that. The more powerful the insights, the more drug targets that will come out of it. I think the other piece is that being able to actually take a genetics first approach to disease is, I think, a concept that will also be very, very valuable, where that an individual carries a certain gene mutation and a certain risk from birth. And in some cases, that may be a mutation that leads to something in childhood, or it could be something that leads to a disease in adulthood. But from the beginning of that child's life what their risks are, and the potential to intervene is there. And this is actually one of the goals of Our Future Health, is to be able to use the data not just to help find new targets, but to help evolve the healthcare system such that you're heading disease off and you're taking burden off of the NHS in the UK, for example, because you're actually you're treating patients early before they really get very, very sick. And so that's kind of the ultimate goal. And so I think with these efforts, we're making progress towards that. And of course, at Alnylam, we're very excited to be part of those collaborations.
Paul, let me ask you just one last question then. We've got gene editing, we've got mRNA therapies. All of this is an effort towards more precision medicines. Where does RNAi fit into that?
Well, I think all of these technologies have promise for patients. Some are more mature than others. And actually, RNAi, we used to be the cool kids that were with the new technology, but of all the things you mentioned, we're really the most mature of the technologies that are out there. We have multiple products approved, the technology's established. And there is really now a point where it's ready to scale. And that's why you see that the pipeline is rapidly expanding across rare and common diseases. But I think all of these other technologies are on their own journey. They all have specific use cases where it just makes more sense to use, or maybe just RNAi is not something that would work in that particular case. So, for example, you've got loss of a gene that leads to a disease, and the best approach there would be to fix that gene. We talked about Rett syndrome earlier on. It's a great example, right? That's not something you can't knock down that gene and cure the disease. You have to fix it somehow. So I think we'll find that we're ultimately going to end up with a suite of really amazing modalities. And it's like science fiction when you think about it. You have to pinch yourself sometimes that this is actually happening and this is a reality. But being able to actually say, okay, this is the disease. What's the right tool in my toolbox to be able to go out there and tackle it? RNAi will play a really big role in that. But these other technologies, they're all going to have very specific areas where those are the right approaches. So very exciting to see what the future holds.
Paul, thanks for joining us today on Beyond Biotech.
Great. Thank you, Dylan.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.